Okay, so we're going to continue on with genetics section 1.14 now. So to start off, we're just going to go through some of the review questions from the last lesson. This was in our last lesson, and we figured out by using the chart that CAU, for example, was histidine, UGA is arginine, AUG is methionine, and this is our start codon. So that is the only code for methionine. There are no multiple amino acids multiple codes for the methionine amino acid. GCC is alanine. GAC is aspartic acid. And then AAA is lysine. Okay, so our next two review questions were, is it possible for more than one codon to give the same amino acid? And the answer for this is yes. So there are, remember, 64 possible combinations of bases and only 20 amino acids. So multiple codons, you'll see for most of the amino acids except AUG, which is methionine. And then what are the three codes for stop? So we have UAA, UGA, and UAG. So remember that the stop codon has no amino acid associated with it. So basically it tells the ribosome it's done since there's no more amino acids to add. So this then allows the chain to be cut and the amino acid chain, which is your protein, to be released. Okay, so let's talk through this next slide. So during translation, the written code, codons, on mRNA is translated into a specific amino acid sequence by what is called transfer ribonucleic acid, shortened for tRNA, in the cytoplasm. This process begins with AUG, which again is our start codon. A tRNA molecule is a small piece of RNA that has an amino acid attached to it. So let's look here. So here you can see the amino acid attachment site. So in our top image here that's shown is what tRNA actually looks like and how it's folded. So RNA is actually always a single-stranded piece that then folds onto itself to make it appear as though it's double-stranded. So if you look at a portion of the tRNA, it looks like a double helix, but it's actually just folded on itself in a loop, and bases complementary pair with each other. So the amino acid attachment site is where the amino acid gets attached in the cytoplasm. The anticodon is on the opposite side, so we can see here it's shown this part here. So this is what complementary base pairs with the codon on the mRNA sequence. So here's just a diagram showing kind of the process of the anticodon pairing with the codon on mRNA. So the tRNA also has a special sequence of bases known as the anticodon, which we just kind of talked about there. So the anticodon is still RNA, so it still involves U instead of T, so this is important to know. And the tRNA anticodon matches with the triplet codon on the mRNA. There is at least one type of tRNA for each of the 20 amino acids. Obviously, again, just like the codon and how there's multiple for one amino acid, this is the same with the tRNA. So here's our amino acid attached to the attachment site. And then you can see here, the anticodon of the tRNA is complementary base paired, meaning it matches to the codon on the mRNA. As the correct amino acids are brought to the ribosome by the tRNAs, they are joined together to form the protein chain that the original DNA coded for. So think of DNA like a set of instructions or a cookbook that tells you what you need. The mRNA is the messenger, and then the tRNA, is what brings our ingredients. So remember that when a stop codon is reached, no amino acid is added. So this tells the ribosome that it's done and that it can stop. So no more, the protein chain is as long as it needs to be. The finished protein chain then leaves the ribosome and moves to the Golgi body, where it is patched up and then it is released as a protein so that it can go on and do whatever its function or job is. So I'm just going to draw your attention to a few things in this diagram off to the side. So the green blob is our ribosome that we can see here. Then we've got our mRNA, and then the tRNAs are kind of shown in yellow. We've got proteins are the kind of baubles being added together, and they've symbolized them by using different colors to show their differences. We can look here and you can see that this section here is the anticodon. And that's on the tRNA. And then you've got your codon, which is the mRNA. And then essentially what's happening is the mRNA strand is being read in a certain direction. So it's feeding through the ribosome this way. And as it's being read, the tRNA molecules are bringing in the appropriate amino acids. So again, the green blob is the ribosome. mRNA is being pulled through the ribosome, allowing the code to be read. And as it is read, the tRNA comes in and brings the amino acid with it. Codons match the anticodon on the tRNA. Each of the colorful circles is an amino acid being added to the growing chain, and chemical bonds are formed between the amino acids as they are being added. Once we hit that stop codon, it stops being read, so the mRNA stops being read, and the protein can then leave the ribosome. All right, so this slide is just a summary of the translation process. mRNA, messenger RNA, brings to the ribosomal unit, which is rRNA, which stands for ribosomal RNA, at the start codon AUG site. A tRNA that is complementary to the start codon binds to the site. In step 2, the next tRNA comes in, bringing another specific amino acid. Step 3, the amino acids bond together to form a chain. Step 4, the empty tRNA leaves the ribosome and a new tRNA comes in. Step 5, the process continues, growing that chain until a stop codon is reached. So then the amino acid chain, which is our kind of starting protein, is released. Okay, so the last thing for you to do here is to work through some review questions on protein synthesis to help you to check your understanding and practice. So there are these sets. Feel free to pause here. We also have a summary slide if you want to pause here, which just breaks down the process of transcription happening in the nucleus and translation happening in the cytoplasm, and kind of gives you an overview of the whole process happening in the cell. You also need to complete this summary of protein synthesis. So there's a bunch of kind of numbers labeling different parts of protein synthesis occurring. So you need to figure out what is happening at each, what's going on in relation to protein synthesis. Okay, and then this slide here is just an overview of what the whole process sort of looks like. So at the top, we have a small section, we have DNA, and then small sections of that DNA are genes. So you can see here, there's a gene here, There's another gene somewhere in here. There's a third gene in here. Different sections of the DNA contain different information for different genes. The DNA, which you see at the top in the box, is double-stranded. It gets unzipped. So below in blue, you can see a single strand of DNA, which acts like a template for the mRNA, which is in red, which then complementary pairs, meaning it matches up essentially with the DNA strand. The diagram provides a nice visual because you can see that the shapes all line up nicely together between the DNA and the mRNA bases. Groups of 3 bases on the mRNA are also labeled, which are your codons. So this is the triplet codon, and translation occurs where the tRNA brings in the amino acid, which comes together to form proteins. So this is just an overview of the whole protein synthesis process, and your labels are on the side here. Okay, last few slides here. So we have officially finished the DNA and protein synthesis section. So that is it for section 1.1. And here is your review for that. So there's lots of practice questions. We'll go through the answers to these on Friday. Okay, and that is the end of this video section.